Ready? Three, two, one. It is started.
Hey, good afternoon, everyone. I really want to welcome everyone here to hear a great talk by Dr. Scott Williams. Dr. Scott joined the Ag Station back in 2000 as a research assistant, and he was promoted to a technician in 2003. And then when he got his PhD at the University of Connecticut, he became a scientist in 2008. So Scott is the Omar Bradley, who's worked himself through the rank. He became an associate scientist in 2014 and a full agricultural science just last year, just about a year and a week ago. Does an incredible amount of research. Currently, right now, what he's doing is he's focusing on evaluating methods for suppressing tick populations in suburban landscapes, looking at the effectiveness of rodent-targeted vaccine for Lyme borreliosis. He's very involved in the active tick surveillance, Lone Star tick monitoring control, and impacts of deer browsing on forest vegetation. Lastly, he's also an adjunct professor in the Department of Natural Resources in the Environment at the University of Connecticut. So let's all give a great big virtual thank you to Scott, and take it away, Dr. Scott.
All right, thank you, Jeff. What I'm going to talk to you about today is a project that Dr. Stafford is the PI on, but that we've been coordinating the field work and doing the research and data analysis and so forth on. And I thought I'd share with you some of the processes that we go through in gathering the data and some of the nuances and what we do day to day in the summer. And also share with you some of the results we've gotten we're in year 4 of this, the 5th year of this study. So one more field season. So as I mentioned, Dr. Stafford's my collaborator, as is Dr. Megan Linske on the project, and we are invaluably assisted by Mike Short, Heidi Stuber, and Jamie Cantoni on this project. And the long sciencey title is Suppression of Vector Tick Populations in a Suburban Landscape through Integrated Use of Host-Targeted and Non-Host Targeted Tick Control Measures. I will explain to you, what that all means, but excuse me. But first, this is sort of the why of what we do, and anyone on this talk is probably well aware of the Lyme disease statistics in the Northeast and upper Midwest in the United States. The graph over here, you can see Lyme disease cases from 1998 through 2018, with 2017 being the highest year on record for reported tick-borne illnesses in the United States. So you can see it's exponential growth. This started way back in the mid-70s when Lyme disease was quote-unquote discovered in Lyme, its namesake. And then on the top right, you can see the map of the United States. And there's one blue dot for every reported case of Lyme disease in each county. And that doesn't mean the case was obtained there. It was just diagnosed in that county. So you can see the heavy presence in the upper mid-Atlantic, New England, southern New England, and the upper Midwest. Massachusetts is anomalous now because they don't report the results, but obviously if they did, Massachusetts would be all blue, like Connecticut and Rhode Island. And then you see some vacant areas in the Adirondacks, Northeast Kingdom, Vermont, Northern New Hampshire, and Northern Maine. Coincidentally, maybe not, that's where you see very low densities of white-tailed deer populations. So obviously with this research, we're trying to, this is valid because we want to solve this problem and not get people sick. Another newly emerging and more frequently emerging pathogen and diagnosis is babesiosis in humans, caused by the pathogen Babesia microti. Similarly, on the bottom left, you can see exponential growth just in a shorter period than Lyme disease from 2011 to 2018. Number of cases obviously is much lower than Lyme disease, but we're seeing similar emerging pattern, and we're seeing it with more frequency on the landscape. And on the right-hand side, again, with a map of the United States, you can see the counties highlighted where varying levels of cases of babesiosis have been reported. And if you look in Connecticut, Rhode Island, Cape and the Islands, that's the heaviest areas report, and seems to be starting at the same focal point where Lyme disease started, and we will expect to see it radiate outward in a similar pattern. And then you see that upper Midwest cohort as well. And the third biggie is anaplasmosis, caused by the pathogen Anaplasma PHAGOCYTOPHILUM. And again, very clear exponential growth pattern through here. A little more cases than Babesia, obviously a lot fewer than Lyme, but it's a very similar pattern. So these are the three biggies, and we also get some others popping up periodically. And but any case of tick-borne illness is one case too many. So we're trying to figure out ways to break the cycle, to break the tick life cycle, the pathogen transfer, and so on. So again, those of you on the call are probably familiar with this, but now I have to go through some of the ecology of the tick life cycle. And in this case, this is our local deer tick or black-legged tick, Ixodes scapularis. So in the center here, we have the adult females lay eggs in the spring, and these eggs hatch into larvae. The larvae then attach to this cohort of animals in the upper right, birds, medium-sized mammals like raccoons and foxes, deer, humans, rodents, if I didn't already say that. And then once they obtain a blood meal, they fall off, they molt, and they become the next life stage, which is nymphs here. So they overwinter in that first year as the nymph, and then that nymph emerges in the spring and gets a blood meal from the same cohort of species: birds, rodents, medium-sized mammals, deer, humans. There isn't a size requirement here for this stage. Then once the nymphs get a successful blood meal, they fall off, they molt, and they become adults. And then the adult female then feeds, and they do have a size requirement. They need a larger animal. So you can see over here on the left that the rodent and bird component fall off, and that's the larger size, medium-sized mammals and large mammals and deer. And in our area, research has shown that 90% of adult females get a blood meal from white-tailed deer. So their abundances are highly correlated with tick abundances, and there's plenty of research to back that. Then the engorged adult female falls off, lays eggs, and the cycle repeats itself. So this is specific to black-legged ticks. There's other ticks in our area, but we're focusing on this because this is the one that's responsible for the most pathogen transfer. And different ticks have different life cycles and different host requirements, but this is complex enough in this one species, so we're just going to stick to this one throughout this talk. Pathogen transfer is a little bit different, but goes along with the same life cycle of the tick. Over here, the adult females lay eggs. When the eggs hatch, they hatch into larvae, which are uninfected, haven't had a chance to feed to take a pathogen. So they're uninfected, and then they feed on that first cohort of animals. And if it's a reservoir competent animal like a white-footed mouse or chipmunk or something, or a shrew or a vole or something along that class, they have the opportunity to take up any of those three pathogens I mentioned previously or others on that first blood meal. Then they become nymphs, and they have about 25% are infected with Borrelia at this point, or lower, but that's a high end. And then they have a chance to feed on humans, on other animals, and they either, if they didn't get infected, have the opportunity to get infected on this blood meal, or if they are infected, have the opportunity to infect their host that they're feeding upon. So it goes both ways at this point. And then once the nymphs get that successful blood meal and either did infecting or got infected themselves, or maybe they didn't get infected at all, they fall off, molt, and become adults. And the adult females typically are around 50% that we're seeing with Borrelia. And I keep mentioning Borrelia because that's the one of concern, because there's so many human cases. But we're seeing a surprising amount of Babesia as well. And then those adults, as we mentioned, feed on deer, and deer are a dead-end host for the pathogen, so they don't get infected and they don't infect ticks that feed upon them with very many pathogens. So this is specific to this tick. So this is other ticks can pick up other pathogens from deer, but again, that's a little too complex for this talk. So the way we go about this is an integrated approach. Everyone wants that one, as we call silver bullet management strategy that's going to wipe out ticks or the pathogen, but it really doesn't work that way because their life cycle is so complex and nature has a way of figuring things out. So if you remove one host component, they simply find another and you're dealing with a different dynamic. So an integrated strategy is better. We're using multiple points within that transmission cycle or that life cycle to try to interrupt it and make fewer ticks on the landscape and fewer sick people. So from a human component, we can educate folks and help them, make them change their behaviors or just be conscious of the fact, you know, when ticks are out and doing some landscaping changes and so forth, or just doing a tick check. They can, personal protective measures, they could tuck their pants into their socks, or like I said, do a tick check when they get back, or wear treated clothing or something of that nature. Landscape modifications, you can see in the top right, Kirby's photo, if you do a mulch barrier there, the ticks, it helps prevent them from migrating onto your property, but of course animals can cross that mulch barrier pretty easy and they can drop off, but it does help. And we've done some research on dense stands of invasive plants and how they're a haven for ticks because of increased humidity levels that the ticks love and thrive in. So if you have dense vegetation on your property, you can manage that. And same goes for leaves. If you get the leaves off your lawn and into the woods a bit distance away from your lawn edge, that helps as well. Chemical controls to kill ticks directly, and there are some natural compounds that have been experimenting with recently that are more repelling than they are insecticides or pesticides. There's biological control efforts. There's host reduction or exclusion. This is pretty specific to white-tailed deer. While there's a lot on the landscape, there's a lot fewer deer than there are mice and other hosts, and they are the major hosts. So we've looked at deer removals or deer exclusion through fencing or something, and that does impact tick populations, but is pretty emotionally charged. And then host targeted acaricides. So is a pesticide that kills the ticks on whichever host we're targeting at the time. And because we know the life cycle, we know when the adult stage is going to be on the deer. We know in spring and fall, so we know that's our opportunity to treat those animals with an acaricide. Because otherwise the ticks are dispersed across the landscape and they're not really concentrated anywhere. And then we've done some vaccine research as well. So this is an integrated approach. These are what we have in the toolbox, and we use these in different combinations. And the recent one I'm going to talk about a little later in the talk, we are looking at chemical control of ticks, biological control, and host targeted acaricide of the ticks. I'll get into this a little bit later. But these are the tools we're using in this project in various combinations. And this is one of Dr. Stafford's slides, but this is the epidemiologic triad. So the interactions between vector, host, and pathogen. This is for ticks and mosquito-borne illnesses. It's similar. So you have host pathogen interactions, and you have pathogen vector interactions, and you have vector host interactions. So if you can interrupt one of those pathways, hopefully you can interrupt pathogen transfer to humans, and it's just trying to figure out the best point to attack for whatever vector host dynamic you're working with. And ways to do this are to control the tick itself through trying to kill ticks, which is what our current study is attempting to do. Or we could control the reproductive host of the tick, that's deer, which a previous study we tried, and I'll talk a little bit about, or control the tick pathogen reservoir host, which is mice, which is difficult because there are so many across the landscape. Or we could reduce pathogen prevalence in the animals, which we could, attempt to vaccinate some of these naive animals. which we've done some published on in the past. And we could do potentially reducing the pathogen transmission from vector to host, and the potential exists there for repellents. We could also alter the environment host, or alter the environmental vector, or change human host behavior or exposure. So these are ways to break this triad and try to break that cycle. And as I mentioned, in our current study, we're just focusing on the vector and trying killing ticks in different ways. But in the past, we've looked at host control, pathogen control. So this is just vector control. Now I can talk about some of the tools that we're using, or some of the management strategies we're using to try to attempt that control of the vector. And when I say four-poster, most of you probably think of this as bed, but no, that's not what we're using. We're using this four-poster. So this is my backside, and my technician Mike in his proper PPE trail camera snapped this photo of us tending a four-poster down in Norwalk on a different project. But that green thing there, this is a hopper that's filled with some corn to attract the deer to hit these rollers that are upright. And you can see Mike is applying an acaricide to the rollers, such that when the deer make contact with it, it rubs onto them. So this is a host targeted approach. So we know this project and photograph was for Lone Star tick control. We know that they're active on the deer in June, but in black-legged ticks it's different. So we actually, Mike's out there just today filling these devices and maintaining them in Guilford for the project that I'm going to talk about, because now is when the adults are on the deer. We use, let's see, permethrin on this. This is a synthetic acaricide that, acaricide is a pesticide that kills arachnids in this subclass, which includes ticks and mites. And again, we're attempting to kill that adult cohort on the ticks to interrupt the life cycle and hopefully reduce the number of ticks on the landscape. So this is how it operates. The deer come in to feed, and they access the trough in various configurations, and hopefully can't help but make contact with that treated paint roller. And here we have some trail camera imagery. These are fun. So I've got a whole bunch here. And I think they took photographs every 30 seconds or so, so it makes for some stop motion, mini stop motion movies. But here's a doe feeding, and you can see her come in, and you can see her head making direct contact with that roller. You can see the roller moving up and down, and then you see her in there getting a good dose and getting right up in there. So you know that's working, and if we've been properly maintaining those rollers there, we can be sure that the doe is getting a dose of permethrin on, that side of her neck, and presumably on the other, and then through grooming, hopefully she can transfer it to the rest of her body. Or we have this little dummy who just chose to stick his head right between the two rollers to feed, which obviously is making great contact with those paint rollers. These are spring loaded. Actually, my technician, Mike Shore, put in a heavier spring, so the deer hit them, they pop back right upright, so there's some give, and it probably helps in treatment. Whereas before, the weaker springs, the deer would put them aside and they would stick parallel to the ground, rendering them virtually useless. And one of our strategies is to put a limited amount of corn in so the deer have an interest in coming to the feeder and checking it out routinely. But what happens is, they come to it and it gets empty, so they're searching for it, and in this case they're desperately looking for corn and moving the feeder right around. So if I had more images, you could see this feeder just kept doing a 360-degree spin as the doe searched for food, and in doing so was making great contact with those paint rollers. Another issue you have with the four posters is non-target species, because you have hundreds of pounds of corn potentially out just sitting in the landscape in that hopper, and the animals know it's in there. And this device is made out of plastic, which is a little bit challenging for the squirrels, but they find their way into them. So here we have a flock of grackles and a handful of squirrels feeding on there, and maybe if they get hit the paint roller incidentally, it'll help them and kill the ticks on them. And then here's another one. We have the squirrel jumps in, jumps out, jumps in for corn, jumps out, jumps in, jumps out, jumps in, jumps out. So a squirrel doing this, you know, every day is going to have some, you know, remove quite a bit of corn that's not available to deer. So again, the squirrel may get treated, but you know, it's not a major host for the ticks, though it is a host. And then other ones, we have, you know, a half dozen turkeys that come in and feed. So issues that go with this, there's some of these animals could potentially restrict usage of the device by the targeted species, the deer. So in this case, we have some interspecific dominance issues. We see a raccoon there feeding in the trough happily, and then this one of our tagged and collared does shows up, and the raccoon notices it and presumably is hissing at this point at the doe, who gets the message and carries on. The raccoon goes back to feeding, and then for good measure makes sure she's on her way. So in this case, the doe didn't want to, you know, interfere with that raccoon, who's clearly making great contact with that paint roller. So he's being treated, he or she is being treated as well, but is, you know, limiting the targeted host from going there. So that doe probably will come back a little later and check to see if the raccoon is gone and feed there. But coming back to this is, again, this is down in Norwalk, a different project, but look at this doe. You can see her ears are laden with engorged Lone Star ticks, and her neck here where the collar is exposed to the skin. So we're writing this project up currently, and what's advantageous about the Lone Star ticks is they feed directly on the ears. The majority of them do. So when the deer is feeding, their ears are basically touching those rollers, so the ticks are getting hit directly with the acaricide. So we're seeing some excellent kill of the 4 posters just with the Lone Star ticks, whereas the black-legged ticks feed, you know, on the ears and throughout the groin area and neck and throughout the body of the animal. So it tends to be lesser effective than Lone Stars. And then, you know, if you're a doe trying to feed here and you're getting karate chopped by a gray squirrel, it's probably going to send you on your way. So anyway, so these are some of the issues here with putting a bunch of corn out on the landscape. Between species, and then within species, you also have some dominance issues at the feeder because of that food source. You have a big dumb buck who could potentially just keep other animals away and dominate that food source, or you could have a dominant doe. That doe on the left, she's got her ears pinned back and she's coming after what could probably be her niece and teaching her a lesson, saying, you know, this is my feeder. So or you have another big dumb buck who's just sitting there, eats and just and chases away any other does or any other bucks that try to come to feed and get treated. So these animals obviously don't know the point of the corn is to attract them to get dosed with this chemical. They're just, you know, think it's a food source that's going to help them through the winter. Or in this case, you have a very aggressive doe who's coming down, probably her niece there. It's dark and doesn't realize she's about to get a hoof to the chin. But I wouldn't want to feed there if that were happening to me. So anyway, we're taking some strategies to try to avoid this. We're purposely limiting the amount of corn that goes in there. So it's not a constant food source, but a sporadic food source, because the point isn't to feed the animals, it's to entice them to get contact with that paint roller maybe once or twice a week to get treated. So we're looking at that and seeing if that can be more effective than keeping 200 pounds of corn in there at all times, which can lead to these dominance issues and lessen the effectiveness. All right, so that was the 4 poster that treats deer. And this tick box here is the same concept as the 4th poster, just on a smaller scale. Excuse me. So this is targeted for rodents, and this is fipronil. Is a very low dose of the compound fipronil, which is the active ingredient in Frontline that you give your pets. The Frontline in your dogs and cats, I think, is around 10 or 12% fipronil, maybe 8 or 9, something along those lines. And this is 0.7% in these boxes that are treating the mice and the chipmunks. So we know that the juvenile stages of the tick, again, with the 4 poster, we're targeting adults. And then this bait box, we're targeting the immature stages. So we know the nymphs are active in May and June, and we know that larvae come out in August. So if we have these devices out there and the rodents are using them and getting treated with fipronil, instead of propagating ticks, they are tick killers. So they're going and vacuuming up ticks who then feed on them and then get killed. So you can see, on the top left here, you have this plastic box out in the landscape, and this mouse is full of ticks. And as you got in here and we break away that top, you can see that there are these non-toxic bait blocks on either side of the box on the interior of it that are attractive to the mice and chipmunks that go in. And then going through here, down here, lower left in the corner, in image 3, you can see this gray wick here, and the mice are forced to go through that and make contact with it, and in doing so get that dose of fipronil. And then as they leave, they've been dosed and effectively frontline, so the ticks that then try to feed on them, in theory, die. Again, same concept, different species cohort and different life stage cohort than the 4-poster. Is great in practice, but when you put a plastic box full of food out in the landscape, the squirrels chew right through it and go right bypass that system and just pull the bait block right out. So now we have to put a heavy metal shroud over this plastic box. And while it can be effective, that whole process is almost cost prohibitive at this point. And the strategy here for bait box distribution is about every 20 to 30 meters around the periphery of the yard. As you can see here, that lawn woodland edge is where you find the majority of your ticks. That Dr. Stafford and Magdarelli years ago discovered that I think it was 80 to 85% of the ticks can be found in this zone. So it makes sense to target that with your treatments in residential properties. And on the right, you can see a graph just showing and some of the research they've done with these boxes, how they can be effective in reducing both total nymphs and the infected nymph cohort with some of these various pathogens. So, all right, so the two, again, now we've had 4 posters with the deer, bait boxes with the mice, and now this is our biological control. This is Metarhizium anisophili. It's a naturally occurring soil-borne fungus. So it's found in nature. It's just been concentrated for this purpose, and it's a fungus, as I mentioned. And the conidia contact the arthropod cuticle and adhere to it. And once they adhere to it, they germinate. The hyphae then penetrate that cuticle, and the fungus develops internally in the host, hopefully tick, and it ultimately overwhelms and kills the tick here. So you can see this female deer tick here has been overwhelmed by it, and it's been killed. It's more selective than synthetic pyrethroids, which kill pretty much any insect out there. This one is specific to arthropods with a cuticle. So if we're spraying it in that zone where the lawn edge meets the wooded edge, we know that's where the majority of our ticks are found, so we're being much more specific. And this is, again, pollinator friendly, and is sold under the trade name is Met52. It's virtually organic, except for the fact they had to add petroleum distillates to the formulation to keep the spores in suspension. It's often used in the greenhouse industry, and Dr. Stafford and with others have published quite a bit of papers on its effectiveness. And again, it can be effective. It needs to get out on the landscape ahead of the peak of the nymphal season, which is difficult to predict because it itself needs to multiply before it can be effective in killing ticks, and then it has a limited lifespan out there on the landscape. So they have had trouble producing this stuff. It's sold. The companies have sold it back and forth, and it's been difficult to get the past couple of years, and the last word we got was that they're not going to produce it anymore, and we have 1 year of treatment, so we're going to have to figure out an alternative treatment here. So these integrated approaches use, as we mentioned, these different strategies, and Dr. Stafford again has published on some of the work that's been done on those strategies. The first of which is, and not the first of which, but one of the more recent ones was in New Jersey, looking at a similar four-poster, the bait box and pesticide applications in 2003 through 2005, and that had 90% control of ticks. And then we did a CDC-funded study down in Reading from 2012 to 2016, where we looked at the bait box, MET52, and deer reduction as integrated strategies. And we were successful in reducing the encounter rate by 2/3 with an infected tick, but there were other issues with that study that I might get into today. The deer component was very controversial, and we reduced deer, but not to the densities we had hoped, and that has some unforeseen ramifications that altered some things. And then this current one here, the 2017 to 2021 USDA funded study, we're partnering with the USDA, who are doing a duplicate study in Maryland with a four-poster bait box and MET52 in various combinations. The Carey Institute is doing a large-scale ITM study in Dutchess County, New York, and Western Connecticut State University is doing a bait box and a synthetic spray study in Ridgefield, Connecticut, and I think they may be wrapping that up. That might be coming to a close. But anyhow, these are some of the ones that have been going on. You can read more about it in this publication here if you're interested, or you can contact me later. But right now I'm going to have a couple slides talking about our integrated approach in Reading. We put out 2 papers looking at the effect on host-seeking ticks and the effect on parasitizing ticks on white-footed mice in residential settings. So here we had 4 1 square mile neighborhoods. One was a control in which we did nothing. One we did deer removal only. One we had the MET52 and bait box combination on 13 different houses. And one neighborhood we had the combination of all 3, deer removal, MET52, and bait box in various combinations. So here in this picture here in the middle to the right of the map on the bottom, you can see the bait box. That's the metal shroud I was talking about earlier. As I mentioned, the deer removal aspect of this project was really controversial, and it was the hunters who actually got in and made a big stink and had to shut it down basically because of safety issues. So as I mentioned, we attempted to reduce them to get them to very low abundances, but we failed to do so. But we did reduce them pretty substantially, so that's had some impact. So this graph here is the juvenile ticks parasitizing mice, the 3-year mean. So fortunately, control was the highest. Deer removal was the second highest, not significantly different than control because we only removed some. And then all 3 in combination was a little more effective, but that includes a deer removal component. And then when we just put the bait boxes and MET52 out, that had the biggest effect. And the reason that's so, because when you remove deer that were previously on the landscape, the tick cohort is still out there, but there's sort of a vacuum of hosts. So they refocus and they start parasitizing on mice rather, and rodents in the absence of those deer. So you see, if you reduce the deer down 50% of what they were, you see an increase in parasitizing ticks feeding on mice in that area, in that vacuum. So this was interesting. So with no host manipulation, just the bait box and MET52 spray, we had the most control. And then going back to that other chart, now I'm going to talk about this USDA study that we're doing in partnership with Maryland. And it's newish, I say, because we're in the 4th of a 5-year study. And we're doing this in Guilford, and this occupies quite a bit of our time. We were sick of getting on the Parkway and driving to Reading every day, 45 minutes or an hour and a half ride, depending on traffic, so we thought we'd do something a little more local. So in Guilford, we have, which happens to be my hometown, we have 7 neighborhoods we identified, and we have 3 rather different treatment combinations of bait box, and all of them get them. So the 2 combinations is bait box 4-poster, bait box MET52, or all 3. and we have 2 neighborhoods assigned to each. So in those areas, we're tick sampling on 83 houses, on 83 properties, and we're rodent sampling on 9 properties at each. And of these 9, they receive the spray, the bait box, and the deer remove, and the 4-poster in combination. And then we're trapping rodents and dragging all these houses here. Let's see, the remaining 20 that we're not treating directly, we're seeing if they have any sort of beneficial side effects from being a part of the study but not being treated directly, because a 4-poster could have area-wide impacts, whereas a bait box is just going to have impacts on the property that it's put out on. So it's sort of a side investigation there. So summer of 2017 was our baseline year where we established the project. We put out the treatment assignments, but we didn't do any treatments. We just trapped mice, sampled ticks to try to figure out what was there initially and what we're dealing with. And then in spring of 2018, we deployed the 4-posters when the adult ticks got active in the spring, and then through summer. And we deployed the 4-posters in spring 2018, 2019, 2020. And then in the summer 2018 through 2020, we did a full implementation of treatments, spraying the Met 52 and putting out the bait boxes and all these properties. And then in fall 2018, 19, and 2020, we did a second deployment of the 4-posters. As I mentioned, Mike Short's out there tending to them right now. Guilford's a very long town, north to south, so we do a fair amount of driving within town. So we randomly assign the treatments to the various neighborhoods by literally a throw of the dice or a flip of a coin. But you can see here these neighborhoods are adjacent to some of these large open space areas. So this is a large piece of town open space here. Here's another large block of town open space, and this is mimicking what they're doing down in Maryland, where they have dense residential settlements and a park that's forested, and that's basically the only open space. So they're working within that park. So while we're not as densely residential as they are there, we tried to mimic their study protocol as best we could. So neighborhood 1, this light green here is Guilford Land Trust property. And let's see, the blue house properties received a letter asking intent if they wanted to join. The pink properties agreed and we're actively sampling on them, and the pink properties with the black outline are the ones that are receiving the full treatment and we're doing the full rodent sampling on. So for instance, this one's involved, but we're not treating their properties with the bait boxes, and we're just doing tick sampling there to see if there's any effect. Neighborhood 2, again, this is that dark green over here to the right is that large piece of town open space I mentioned, and this one's getting the 4-posters. So the 4-poster locations here are with these stars, and given the tick density, we estimated that each 4-poster would cover about 50 acres. So what we did in GIS was take a 50-acre circle and made sure that all the houses that we were treating, and put a 50-acre circle around each 4-poster location and made sure that all our treatment houses fell within that 150 acre treatment zone. And if it didn't, we strategically moved the feeders around to try to accommodate that. Another neighborhood here, this is south of that large property I mentioned, similar distributions. You have clustered houses who agree to it, and then you have these 2 sort of satellite houses. It's difficult to get them all within the same area just because landowners, can opt in or opt out at their will. And then we have to adjust our 4-poster locations accordingly here. Similar here, large piece of open space here, 4-poster here, here, and here, and that covers all of our treated properties. This is our control property. Of course, we had the most interest. This is down in Sachems Head, down near the Sound. And we had a lot of participation down here. I think we had an eager participant who was recruiting neighbors. And of course, by a flip of the coin, this wound up being our control. And some of the people down there aren't too happy about that. This one, similar here, you can see the 7 properties here around that circle here all clustered together, which is what you want to see ideally, because if you're treating on one property, you could have bleed over onto the other, and if you're treating on that property too, you're going to have double the effect. So we're seeing pretty good results here because these 7 are clustered together. And then again, we have these 2 satellite properties up there. And then this one is sort of southern. This red is state property, and these are kind of scattered throughout. So we put out 10 of the bait boxes at each of the 9 properties within each of the 6 treatment neighborhoods, or 540 boxes total. And then when they get used up in, say, July or so, we take them out and replace them with new boxes in anticipation of the larval season coming in August. And then it was difficult to find an applicator to come out and spray these small study area zones with their big rigs. So for about three-quarters of the price we pay them to do one application, we bought this 100-gallon spray rig, and Mike and my staff have done the applications ourselves in May, June 2018, 2019, and 2020. This is at the farm. If anyone wants to use it in their near future, let me know, and you're more than welcome to use it. It's quite handy, and it's in great shape. So again, we deployed the 4 posters in spring and fall. I've been through all this. And we put 50 composite corn in each week, and you can see this little buck feeding and pawing at the 4 poster, and in the next picture he tips it over on its side. So we purposely limited the amount of corn in there to pique the deer's interest, to make sure they kept coming back. And then one day it's full with another 50 pounds, and there's a intense interest in the corn, and they get treated, and then that interest wanes, and then they come back periodically and hopefully get treated with those. Again, the point of the corn isn't to feed the animals, it is to entice them to make contact with the roller. This is a picture of Mike doing the tick sampling, but as I mentioned here, despite that photo, Heidi Stuber is the one responsible for coordinating all the tick dragging efforts throughout the summer, and her summer staff on 83 properties, dragging them multiple times. That's a huge effort, and I just want to give Heidi a shout out. And she established permanent transects of known distance on the lawn edge, as on that zone I mentioned previously, and also a wooded area on the properties that was conducive. And she sampled, and her crew sampled all through May and June and into July, and we captured that peak nymphal season, and you can see it tail off through June. Minimally, each house was sampled 8 times. And then we sent the ticks off to CDC to be tested for pathogen presence, and we got some good data there. And the results I'm about to talk to, we just recently sent the 2020 samples off for testing, so we don't have the pathogen results yet. We just have them from treatment year 1 and 2, or 2018, 2019. And then my crew's component was the rodent trapping, and we trapped them late May through September when the nights are warm enough for the mice to survive being in an aluminum trap. Put 12 traps out per house, and we ear tag them, blood sample them, ear tissue sample, weight, we quantify the tick load, and we remove any engorged tick like pictured here, and we bring it back to the lab. We've got a bunch of non-targets: chipmunks, opossums, skunks, flying squirrels, snakes, birds, ermine, weasels, all kinds of stuff. But we released those, and I think something like 97 or 95% of all of our captures are white-footed mice, which are the major reservoir for these various pathogens. So they're very important here. So we trapped each of the 63 homes 4 times in 2017 to 19, and 5 times this year. And this is a lot of numbers here, but you can see the acreage Heidi sampled for ticks and her crew, between 13 and almost 17 acres solid of coverage of drag area, which is no small task. And then, resulting in anywhere from 150 to 250 nymphs per season, or 800 in total. And as Kirby mentions, as you're trying to kill these ticks, you're limiting how many you can sample which gets into issues with your statistics because you have such small sample size. Because if you're effective, you're not getting the return on the samples that you're out there trying to obtain. And then as far as mice, we sampled anywhere from 220 to almost 600 unique individuals. And some of those you recapture several times. So between 300 to 800 total captures. You know, we're looking at 1,500 unique mice and 2,200 captures. So this is no small effort on our part. And, you know, these are pretty good, robust numbers across these 7 neighborhoods. So we got pretty good sample sizes on this. So initially we're looking at the results, and we got, you know, very excited here. We were super excited that our control was 4.2 juvenile ticks parasitizing each mouse, and then in our treatment areas, it was 8 times lower than that, and significantly different. But unfortunately, that was from our baseline year, which was we had assigned the treatments, but hadn't applied any of them. So basically, what we found out was our control neighborhood down there in Shoreline, Guilford, had very high abundances of ticks that we had to deal with, which was unfortunate. So over here in the lower left in the 2017 baseline year, you can see those data I just presented. And then you can see in 2018 in treatment year 1, the control grew. And then in year 2, it grew even more to about 10 ticks parasitizing each mouse we captured. And then in this past year, it dropped back down to 3.2. So you can see our treatments there, the yellow, gray, and maroonish bars were much lower than the control. But to what effect, our treatments had, it's been a little challenging to tease out in these preliminary data analyses. So this is unfortunate that our control was so high. We had so many ticks in the control neighborhood, and then it plummeted and dropped down in 2022 to a third of what it had been previously. So as I mentioned, it's unfortunate that it's had such a high abundance, but that was just a roll of the dice, and that it had such an annual variation in ticks, but it wasn't a deal breaker. So we have to assume across all of our sites that environmental factors such as precipitation, lack of precipitation, humidity, temperature was having an equal influence on all of them. So, and that's safe to say, seeing as all these neighborhoods were in the same town, only a few miles apart. So in order to standardize the treatment influence, we looked at percent change for each year, for each treatment year as compared to the baseline year for each treatment in this study. So just looking at how it changed from the baseline year to treatment year 1, 2, and 3. So for instance, if we look over here at the control from the baseline year to treatment year 1, the control population of ticks parasitizing mice grew almost twice, almost doubled. And then in year 2, it more than doubled. So you have to assume that if we're not doing any treatment there, that we're seeing an equal growth on the rest of the properties that we are treating. So for instance, where we're not treating, if it grew by 95% in treatment from treatment year baseline to treatment year 1, where we're doing the bait box and MET52 combination, it only grew 10%. And where we're doing the 4-poster and bait box in combination, not only did it not grow by 95%, but it decreased by 39% from the baseline year. And when we're doing all 3 in combination, it didn't grow 95, but decreased by 83%. So we're having an impact here despite this annual fluctuation in the control. And if we look at baseline compared to treatment year 3 for the control, that went down 22%, but our treatments went down anywhere from 88 to 96%. So we're having almost a 4-fold influence on the ticks parasitizing mice, which is good news. And again, it's encouraging to see this here, and this was just sort of a preliminary analysis for this talk, and we've got to get into the details a little bit more. I'm not going to deal with that now. And then we looked at a percent of mice infected with Borrelia, and for the control it was great. We had equal for years, baseline year 1, year 2, all about 48–50% infection. And then for our treatments, with the bait box MET52, We're seeing it decrease from 42% down to 20% in 2019, which is treatment year 2. Similarly, for the 4-poster bait box combination and for all 3 in combination. So we're seeing treatment year 2 percent infection with Borrelia significantly lower than both the treatment year 1 and the baseline year. So this is very encouraging. As I mentioned, we don't have the data from this year, but I'm looking forward to getting it, and hopefully we see this decreasing trend in infection in mice, meaning that we're controlling the ticks enough that they're not reinfecting the mice. And hopefully this year and next year we'll see that continue to decline, and hopefully we can have a bunch of Borrelia-free mice on the landscape and keep that knocked down. So this is quite encouraging from a public health standpoint. And then this is host-seeking nymphs per 100 square meters on the lawn, the data that Heidi obtained. And unfortunately, this is a bit wonky and hard to interpret because it looks like these red bars are our control. It looks like if you do nothing, you get significantly greater control. But again, that's just the environmental fluctuation. You can see each year is sort of a scaled down version of the previous year. So these are, we're not seeing much variation between years within treatment areas. So we have some work to do here. So this is a little discouraging versus the parasitizing tick loads, which was less discouraging. And this is that same where you're looking at the baseline year to the treatment years 1, 2, and 3, and that control, you can see it appears that we have really good control in year 3, and that's just environmental fluctuation. And then the 4-poster and bait box, it seems as though we're increasing the amount of questing ticks on the landscape. So this isn't great. Hopefully we'll get some better data this coming year, and hopefully Maryland has some data that can counteract these data, but we'll see. And this is the density of Borrelia infected host-seeking nymphs on the lawn. A little more encouraging than just overall abundance. Again, you can see there's quite a few infected ticks in our control area, and those stay moderately the same throughout the 3 years, and then we're seeing some impact from our treatments. Unfortunately, all 3 in combination tend to be the lowest, so that's somewhat encouraging. So the take-home message here, the results are, well, a little bit murky, and we're teasing through them, and we're not sure what to attribute it from. It could be some extreme weather we've been having, whether it be drought that does not favor ticks, or mild winters that do favor ticks, or maybe there's enough, coastal Guilford is enough of a microclimate compared to northern Guilford that, you're seeing some variation there in sites. And it has been proven difficult to detect a difference in tick reduction in both the host-seeking, more so in the host-seeking than in the parasitizing populations. But again, that Borrelia reduction in infection in the mice in all 3 treatments is showing us a promise. So we're looking forward to getting some more data there. And as I mentioned, hopefully in year 5, this coming year, things will be clearer, and we'll be able to synthesize and put these all together and come up with a more cohesive story. We'll have all the infection data, and we can, hopefully this is just a little blip, and we'll have a better story to tell after this sampling year. So I've mentioned our research team, Megan, Kirby, myself, and Mike and Heidi and Jamie. We couldn't do it without them, and the summer workers have been great. And my dog in the photo seems to be showing us what he thinks of our study. But anyhow, that's all I've got. If we have people from offsite who want to contact me, here's my contact information. And thank you. I wasn't sure how this was going to go on this platform, but I'd like to say I think it went pretty well. So if you have any questions, I think we have 10 minutes or so. So what do we got? Is a tick box available commercially? It is, but you need to have a licensed applicator put it out because it contains that fipronil. So there's a company, Connecticut Tick Control out of Norwalk. can do that, and you can contact them or contact me, and we can put you in touch with them. But I think they're the sole supplier for the tick box. Let's see. Why were the tick numbers and pathogens so high in 2017 compared to other years? Was that, I assume this is in reference to the total US cases or in our data? I'm not sure. In either case, I'm not quite sure. It can depend on reporting rates, or it could depend on acorn abundance, actually. So when you have high acorn years, the year after that, you have high rodent years, and then you tend to have high tick years, because with high rodent abundances, you tend to give the ticks a better chance of successfully feeding the younger ticks, successfully feeding, moving on to the various life stages. So it could have been a high acorn year in 2016, and then you see a boom in ticks thereafter and disease follows. We've done some anecdotal research on that, and it's a real thing that acorns really do drive mouse abundances and in turn tick abundances, but we haven't taken it to that next step to look at disease cases in humans. To be successful in the long run, do you anticipate the tick control year after year after year? Yes, unfortunately, yes. And most of these studies are 3-year studies, and you know, with a 2-year life cycle of the tick, you're really seeing your full impact just starting in year 3. For us, that was this year. So this is a 5-year study with a baseline year in which we did nothing. So we're gonna have 4 years of treatment. So hopeful, I'm really hopeful that this 4th year, as the tick generation goes on and we're getting further and further from that 2 years that it takes them to reproduce, that we're going to have more success. And then when we stop, we might continue to do some sampling there to see how long it takes for them to recover. Kirby's done some of this, and he knows, I think after a couple of years of non-treatment, it comes back almost to the same point it was before you started treating. If bitten by an infected tick, which of the host species is more likely to get Borrelia infection? Are some species more or less resistant to infection? That's a very good question. And so this goes to reservoir competency. It's the ability of a host to get infected and reinfect ticks. Deer, as I mentioned, can be infected with the spirochete, but it doesn't affect them at all, and they're not able to give it back to the tick. White-footed mice, the pathogens flow freely from tick to mouse and from mouse to tick. They're very competent reservoirs. It goes back and forth, and with chipmunks too, and some of those other smaller rodents. And not much is known on various bird species or medium-sized mammals as to their level of competency, but it's those mice that really drive that. And Megan's done some work on looking at host diversity in the landscape, and if you have a more diverse array of hosts and the ticks are feeding on that diverse array, there's a more likely chance they're going to feed on a reservoir incompetent. So why you may have more ticks on the landscape because you have more hosts, you're going to have a lesser infection in that tick cohort versus a less diverse population with majority mice where the ticks are feeding on that reservoir competent reservoir and getting infected and reinfecting them back and forth. From Jeff, why were the mouse numbers down in 2018 and 2019? Not so sure. Again, I'm guessing 2017 was on the heels of a mast year previously, which is why we saw so many in that baseline year. And then they can't sustain themselves, or we see owls and hawks and things realize there's a ton of mice on the landscape, and they start to switch over and use those as their prey base. And actually that's what happened that year. Jeff just jogged my memories. I don't know if you guys remember, but we're seeing a ton of owls, and social media, people posting barred owls everywhere. We had back-to-back mast years, and then we had a boom, 2-year boom in mouse years. I think 2016 and 17 were those high years. So we had a lot of owls and a lot of birds of prey born to feed on that abundant host reservoir. And then I think they got knocked back when there were so many predators on the landscape feeding on them that and knocked them back in the following 2 years. So it's just classic predator-prey dynamics. And we were able to quantify that a bit and had an article in Connecticut Wildlife about that relationship. From Pete, if a yard is littered with deer droppings, would it be safe to say ticks are likely or certain? Yeah, if your yard is littered with deer droppings, you have a lot of deer, and if you have a lot of deer, you probably have a lot of ticks that they've dropped off or are feeding on them. And so it's this time of year, as I mentioned, if you're seeing a lot of deer poop in your yard, you're probably having a lot of ticks because the ticks are feeding on the deer this time of year. Typically, they spend most of their time in the wooded areas, but at night the deer do come out and feed on residential and foundation plantings and so forth. So there is that opportunity for the ticks to drop off in your lawn or on your back deck, if it were. Let's see. One more question. I said, did equal numbers of animals visit control and treatment for poster/tick boxes? Is there any reason why control sites would be more attractive to animals/ticks? The control sites, we didn't put out for posters or tick boxes. We just sampled mice and dragged for ticks. Is there any reason the control sites would be more attractive to the animals? I think the control site was just down there. It's more dense residential setting. It's more mild because it's adjacent to the sound, and hunting and is virtually a non-entity, and they do have a ton of deer down there. So I think they just have a lot of wildlife, a lot of hosts, not a lot of hunting, and I think that's the reason for the increased tick load down there. And like I said, that was just, we assigned that based on the flip of the dice and weren't all that shocked to find that many ticks down there. Great, I don't see any more questions. Thank you all. Again, I felt this went well, and thank you for your time.